such an honor to be able to talk about my passion. I'll start with development, but I will definitely move on to sex specificity, so bear with me. So 1 in 4 people experience one or more mental illness, and this is the World Health Organization chart of the age of onset of 3 major mental disorders: anxiety, substance use, and mood disorders. And you can tell that they emerge very, very early, and this is the median, so meaning that 50% in anxiety emerges by age of 10, and this is the interquartile range. So these disorders are actually quite neurodevelopmental. And actually, about 10 years ago, National Institute of Mental Health released a statement that actually we stopped finding new therapies because we actually don't look at early life research. So straight to the point, I'm sure you've seen fear conditioning. We do tone shock pairings, and we look at freezing as a behavioral measure. And then in extinction, we repeatedly show the fearful stimulus, and eventually, if it's not followed by any outcome, the fear response or the memory response declines. And this extinction is the fundamental process underlying exposure therapy in many mental disorders, but especially for anxiety disorders. And of course, we test to see whether you remember the conditioning or the extinction memory. So in this case, if the freezing is low, extinction is great. But when the freezing is high, we say the extinction is actually not very good, that there's poor extinction recall. For example, you might feel fantastic in a therapeutic environment with your clinician, but then as soon as you go back home, which is a different context to your exposure therapy, you might feel afraid again. And I love studying this neural circuitry because it is quite well conserved between rodents and humans. And I look at both rodents and humans, which I'll briefly describe. So my lab has started with a developmental focus, initially, characterizing the developmental period before and after, when we can do large-scale spatial abilities. So, you can do ball pit, open field, or navigation in children, and 7 years is when they reliably show, and that's 3 weeks for, Morris Water maze in rats. And then we look at maturation, which is adolescence and adulthood, looking at ages, 10 to 15 years when girls start menstruation and the boys start to show secondary sexual characteristics. And because of the neural circuitry, we say the first part, there's a lot of change, it's actually fundamentally different, development, and second part, it is more refinement and is maturation. Now, my postdoc has given a talk in development, so I'll briefly talk about the maturation that I did not present yesterday. So there's quantitative changes in the neural circuitry. That's why we say this is a refinement period. So our adolescents are a lot like adults, but they just do things a bit less efficiently depending on the cognitive task, especially in inhibitory control. So when we looked at pre-adolescent, adolescent, and adult animals, they fear condition very similarly, and they learn to extinguish very similarly. But when we actually look at the extinction recall, compared to pre-adolescent and adult that show context-specific expression of fear, adolescents actually don't remember extinction at all. And this is actually consistent with the human anecdotal evidence that actually it's not that they don't learn, but they seem to forget the second experience much more rapidly than the first experience. That's why we ourselves remember whatever happened during our adolescence, like the songs we loved or even first love. It's very, very hard to forget. And this behavior was correlating with the activation of mitogen-activated kinase. When in the infralimbic cortex in particular, the adolescent animals, even after extinction, looked basically like the no extinction group. And when we doubled the amount of extinction, actually we could see good extinction recall and subsequently increased level of activation in the prefrontal cortex. But this was all done in males, I have to admit. So this is back in 2011. But hold on to that thought. So recently, we used functional magnetic resonance imaging to be able to translate. So in humans, because I don't trust what they say, so I always measure their sweating levels, their skin conductance response. So the more anxious or more aroused we are, we sweat more and we conduct more electricity in our fingers. So when we look at face paired with scream compared to face never paired with anything, they sweat more and there were no age differences. So this asterisk is not age effect, but that there was a significant conditioning effect within session. But extinction and conditioning, there were no age effects. But when we gave them a break, and then we made them recall the extinction training, there was a significant difference where adults were really good at suppressing their arousal, their sweating after extinction, but adolescents forgot and pretended that they don't remember ever receiving extinction. And the brain, consistent with our expectations, I guess the adults show that medial prefrontal cortex activation with extinction, but adolescents actually did not show that activation in extinction, consistent with the rat results. So we were able to translate that. Yes, we can model, back translate preclinically what happens in the clinic. Now, but females share the most burden for most diseases, not just mental health-wise, but a lot of diseases. So this is an interactive website. So I'm really sorry, I don't know how to make QR code yet, but I really recommend you to check this out. There's so many statistics that move around to really show the female burden. And anxiety is definitely one of them. Even though mental illness is here, they also show specifically for anxiety, which is down here. So, but the paradox has begun. So about maybe I would say only 15 years ago, groups in America started to look at, what's sex-specific about exposure therapy? And what they have shown is that actually high level of estrogen or estradiol in rats is actually good for fear extinction. So if you test the rats when estrogen is high, then they tend to remember extinction better and freeze less. But if you test them at phases where estrogen is low, they actually show a lot of freezing. But this doesn't make any sense, because why is it then that females have more estrogen than males, but then they have more anxiety, right? So, and this is the finding. So when they split the animals from pro-estrus phase, where there's a lot of estrogen, and metaestrogen phase, which there is fewer. I think actually, to be honest, the estrus phase is a bit funny, but yeah, they basically claim this. But the thing is, when you actually look at all the fundamental studies of hormone cycle, which happened in the 70s, and this is, I guess since the 70s, people couldn't find a graduate student that were willing to measure things every 4 hours, 24/7. But anyway, so throughout this cycle, clearly the pro-estrus phase has the most estrogen, and estrus phase has the least estrogen. So these are usually the typical focus right now. So I really recommend you best to group this way rather than metaestrus versus proestrus if you want consistent findings. But I argue that, it's because sex differences in anxiety actually peak in adolescence and also start actually quite early, 6 years of age. I mean, it's a pretty old finding, but at least the adolescent difference is quite consistently shown since in this 90s finding. So firstly, in rats, I decided, okay, there is actually no adolescent rodent data at this time looking at phase and estrogen levels. So we first mapped that out. We, you can do with cytology and of course serum hormone levels. And yes, thankfully, the metaestrus, diestrus, proestrus, estrus phase are consistent with the sex hormone levels observed in adults if we look at adolescents. But what's actually different is that the cycling is absolutely everywhere. So during the onset of puberty, in rats at least, the conversion from, when they're not even menstruating, we call it unopened vagina, they, a lot of them actually haven't even started menstruating from 35 days of age, and the transition to each cycle is actually not very smooth. So sometimes, in adults, you go from met and di, pro-estrus, but adolescents would be like met, pro-est, di. So they were absolutely all over the place. So that was a really, really interesting finding that consistent with the humans, puberty means that your fluctuation in hormone levels are crazy. You're not having these regular cycles like in adults. That was our first finding. And then we tested a lot of rats because I thought I really want to see have a well-powered study so that I look at conditioning, extinction, and test. separated to estrous cycle in our adolescent animals, depending on their cycle on conditioning day, depending on their cycle during extinction day, and depending on their cycle during test day. So the first was when we separated everybody depending on conditioning estrous phase. And there was a minor subtle effect during conditioning, but actually hormone levels or cycling during conditioning had no real impact for the rest of the day, rest of the feed conditioning process. But when we grouped the animals depending on the extinction estrous phase, we saw this effect where the phases associated with higher estrogen, they remembered extinction far less. And the ones where they didn't have much estrogen, they looked just like males. And then when we grouped the groups depending on test hormone phase, then there were no effects once again. So actually, high level of estrogen during adolescence is terrible for feed extinction. And this finally explains female vulnerability to anxiety disorders, not being able to suppress fear, because anxiety disorders happen early in life, and the puberty makes it much, much worse. And we did gonadectomy. Long story short, gonadectomy actually reverses the findings, and we confirmed the phase and gonadectomy with the serum levels of hormone. So actually, not only estrogen is bad for you during puberty for fear suppression, but testosterone is good for you. So looking at the entire, process of, we started to actually now look at sex specificity and hormone-related learning processes in acute fear conditioning, acute fear extinction, and relapse. And this is a rough summary that we actually have. And I have to admit, starting sex-specific research actually feels really late now, because every time I do something, there's some interesting sex finding that actually makes sense and matches with epidemiology, especially because I look at early in life and also looking at pre and perimenopausal. I find that the model is well replicated and it gets cited, so I really recommend definitely doing sex specificity, but it is more work because you need more power to analyze it. Also, my recent Australian Research Council Future Fellowship, where they only give to 100 scientists in the whole country each year, but no field specificity. It can come from arts, it can come from dance. I was able to be selected as one of the 100 scientists that best represent individual field. And that was actually, I was highly commended because I said, I'm going to look at 9 stages in our life, circling around hormone development from really early to postmenopausal. In rats, actually, that wasn't a human project. And I'm going to look at sex specificity, how we learn, because it really applies not just to disorders, but in healthy behavior too. And so that was really highly rated as a really good project to do for 5 years. So it really benefited me. Thank you.